Hello, everyone. Good afternoon, and welcome to today's Congressional Biomedical Research Caucus featuring Dr. Stephen Quake from Stanford University. Before I get to introduce Dr. Quake, I'd like to go through a little bit of caucus business, if you will. First, I'd like to thank the co-chairs of the caucus, Congressman Brian Bilbray from California, Congressman Charlie Dent from Pennsylvania, Congresswoman Jackie Spear from California, and Congressman Rush Holt from New Jersey. They all showed tremendous commitment and dedication and ongoing support for the caucus. I'd also like to thank the Howard Hughes Medical Institute for providing a grant that supports our ability to host these timely and important briefings and to supply you all with lunch. As you can see, we do videotape every briefing. You can find past briefings on the CLS website at coalitionforlifesciences.org. And at that site, you can also register for RSS feeds in order to be alerted for future postings. And if you haven't already, be sure to pick up a schedule of briefings that are on the registration table outside the room. That way you can be alerted to all the future briefings coming up. Okay, so now that business is over, I'd like to introduce Dr. Quake. Dr. Quake is a professor and co-chair of Bioengineering Department at Stanford University. He pioneered the development of the microfluidic large-scale integration. This technology has been used in applications as diverse as discovering a new drug for hepatitis C, mapping the genomes of environmental microbes, and measuring gene expression in individual cancer stem cells. Commercial versions of this microfluidic large-scale integration are now used in hundreds of laboratories around the world for diverse purposes. Dr. Quake demonstrated the first successful single-molecule DNA sequencing technology, which has been commercially developed and is leading candidate to develop the first $1,000 genome. In 2009, he and his two co-workers sequenced his genome using this commercial version of the single-molecule sequencing technology that he developed, an event that was widely reported in the popular press. He has received numerous awards from the NIH, MIT, Forbes, and Popular Science, among others. He is a founder and scientific advisor on the board, scientific advisory board chair of Fluid, I'm going to pronounce this wrong, I apologize, Fluidigm and Helicos Biosciences. So without further ado, please join me in welcoming Dr. Quake.
So thanks for having me here. They told me not to use any scientific terms in my presentation, and that's a bit of a challenge for me, given that science is what I do and what I'm going to talk about, and sort of the equivalent maybe of trying to give a political speech and not say God bless America. You know, it all just comes out reflexively without thinking. So I'm going to try and define the terms, and so it may start on familiar ground for you, and hopefully that will provide some common language. But if you're getting lost, please raise your hand, interrupt, ask questions along the way. I'm fine with that. So I want to sort of use as the starting point this, to frame the talk, this article that Nick Wade wrote in the Times a couple years ago, where he kind of took the Human Genome Project to task and said, hey, you finished the genome 10 years ago, and none of the stuff you promised has really come to pass as far as medical benefits. And so it's a real disappointment. And you know, I really took umbrage at that and thought it wasn't a very accurate sort of portrayal of what the fruits of the Genome Project have been, especially in terms of the medical applications. And so I'm going to, you know, basically the goal of the talk is to try to refute the article, and we can have a little discussion of that at the end if you like. So let's talk about what's the genome, and genome is made up of DNA, and what's DNA? So DNA is this really interesting molecule that is the blueprint for making all the structures in the cell, all the proteins, the cell wall, everything like that. The DNA is the blueprint for making all that. And so it stores information. And what it is it's a big, long molecule, kind of like a floppy piece of spaghetti. And all the DNA in a cell is called its genome, or an organism called its genome. E. coli, which is a bacterium that lives inside your gut, its genome is about 1 millimeter long. So it's a molecule that's very small in diameter, just a few atoms wide, but can be very macroscopic in its length. And you can actually see DNA under a microscope with your eyes. It's really kind of amazing. And the structure of DNA is this famous double helix, which is it's like having two spiral staircases intertwined with each other. And the steps on the staircase are 4 different chemicals that are used to encode the information. And those steps, those chemicals can come in arbitrary order. So it's like the letters in a book. And, and that's how information is stored in DNA. It's chemically there. And this is kind of the mechanism that all of life uses, except for a few viruses. So there's various protein machines that can read this information out. They can copy DNA. They can transcribe it into RNA, which then becomes a template for making the proteins and so forth. And so it really is sort of a fundamental thing. And this sequence information is used for all kinds of practical applications beyond its intrinsic biological use. It's used for forensic purposes, for doing DNA testing, which I'm sure you've heard of, genetic testing for disease, identifying pathogens. If you're sick, it can tell you what kind of pathogen you've got. So it's got all kinds of practical purposes. And it's kind of amazing. It turns out that not only does all of life use DNA as its common alphabet for storing its blueprints, but the language is essentially the same too between all organisms. It's very closely related. And so in sort of one of the really extreme examples, you can look at yeast, right, which is a little microorganism used to make beer and wine. About 30% of the genes in yeast, so a gene is the blueprint for a given protein, about 30% of the yeast genes have some kind of similar gene. There's a similar corresponding gene in humans that is very closely related. And how closely related are they? Well, you can look at the sequence identity. In some cases, as high as 96%, 89%, 75%, 66%. So they share, through evolutionary history, the sequences haven't diverged that much. And there are some really dramatic examples of these relationships. And in some cases, what you can do is you can delete a given gene in yeast, and you can put in the human version of the gene, and the yeast will be fine. That function will be restored. And so it's kind of a very deep connection. And so this is one of the reasons why biologists like to study model systems: yeast, worm, fly, fish. They're very informative because a lot of the biology is shared. And it all comes back to this sort of evolutionary relationship in the DNA sequences that are shared. So, as I mentioned, the genome, the Human Genome Project essentially finished a decade ago. And why are we still sequencing human DNA since the human genome is done? Well, it turns out what the Human Genome Project sequenced was the average human genome, so the parts of the genome that are shared between all of us. And there's parts that we don't share. That's why we're different from each other. We're not all identical twins or clones. And we differ at about the part per thousand level. So you pick me and a random person, another random person, we'd share about 99.9% of our DNA sequences, and that 0.1% is what creates all of human diversity in all its glory. It's kind of amazing. And so you can, for the purpose of this talk, call them mutations, these differences. And as I mentioned, this is the source of human diversity. It's related to causing all kinds of diseases, disease susceptibility. It's related to whether drugs work on you or have side effects. And so measuring this diversity has become sort of a primary goal in the post-genome era, and that's the reason why we're still sequencing, is to try to understand the differences between individuals and how it relates to disease. Does that sort of make sense? Yeah? All right. So my role in this, as Lynn said, I wasn't part of the original Human Genome Project, but in my lab about a decade ago, we demonstrated the first single molecule sequencing, and that led to what was at the time the fastest, cheapest DNA sequencer, and I used it to sequence my genome. And so I was among the first handful of people in the world to see their own genomes. And in trying to figure out what that all meant, I enlisted the aid of a number of colleagues at Stanford Hospital, and they did a clinical annotation of my genome, which was probably the first time that had ever been done for anybody, and tried to sort of answer this question of how does a doctor treat a patient who comes into their office with his genome in hand? And that's what I'll tell you about in the next few minutes. It's worth pointing out that this has been an extraordinarily sort of fertile time for technology development in the genome world. And it's been, you see technological improvements that are even faster and more rapid than in the semiconductor industry. So costs of acquiring DNA sequence are going down by orders of magnitude on almost a year-on-year basis. And the amount of data that's being created is going up enormously, and this sort of counts genomes. This is sort of the rough draft of the Human Genome Project, completed. Craig Venter, Jim Watson, me, and others. And it's piling up at an enormous rate. And it must be said that there are sort of enormous policy implications to this, and one feels like the policy discussion is really lagging the technology and the scientific output. And so there's a bunch of really interesting questions that they're going to come out of this that have to be dealt with in the policy arena.
I have a little trouble reading this.
Yep.
For an individual to get a sequence
Yep.
now.
Yep.
I thought it was typically, a bargain price was $1,000. But
That-
it looks to me like I can't quite read it, but
Yeah.
I think you're suggesting that you can actually perform the sequence for a lot less than that.
Yeah, today, well, no, this is cost per megabase, which they plowed, which is it's a fraction of the genome. Yeah.
Oh, it's
So
per fraction, okay.
people this $1,000 genome is something that people have been aiming for, and we're probably a couple years away from that, I would say right now. Today the costs are between $5,000 and $7,000 for a human genome.
Thank you.
Yeah. And you'll see how rapidly that has moved. So I think I'll skip some of this. This is just sort of illustrates some of the history of how rapidly the single molecule sequencing advanced. And I want to point out the folks who actually sequenced my genome. It's Norma Neff who did all the sequencing. So one person and one machine. And Dmitry Pushkarev was a student who did a lot of the computation on it. And so it was, when we sequenced my genome, it was the first example of being able to sequence a human genome with just one instrument, one person, one lab. You didn't need a whole big genome center and room full of machines and all that. It was a real kind of turning point for the field that kind of democratized access to human genomes. And you can sort of see that on this slide. This was a table I made up when we published my genome, and we compared my genome to all the ones that have been done previously and published. And you can see sort of the rapid technological improvement by any number of metrics. The cost for doing the genome, which is basically the cost of the chemicals, the reagents to do it, you can see that zeros are coming off with every year. And so sort of exponential decline in growth, decline in cost, rather. This was the technology. This was the individual sequenced. This was the number of machine runs that were needed to do the genome, which is kind of an estimate of the capital costs or capital infrastructure required. This was the number of authors on the paper, which was an estimate of the manpower required. And so you can see why this was sort of a turning point in 2009. And nothing stopped there in 2009. It's continued. You can knock off another zero on the reagent cost. This is down to 1. Hard to make that smaller. And it's become a rather routine thing to do now. And many, many people are able to play in the sandbox as a result of this technological advancement. And it's really been good for the field.
What does coverage mean?
Yeah, that's a technical thing. That's how many fold coverage you've over-sequenced the genome. And it's a sort of a metric for completeness, sort of. Yeah, it's a technical thing, but it's not totally relevant for this conversation. Today, coverages are in sort of the 60x range, what people do to feel happy with the genome. And so, what do you get out of it? As I mentioned, you differ at sort of the part per thousand level, and what that means, since there's about 3 billion base pairs in the human genome, that there's about 3 million places where any two individuals will be different. And so we took the other 2 published genomes when mine was done, which was Jim Watson and Craig Venter. And my student Dmitry made this Venn diagram of where we shared SNPs, these mutations, and where we differed. And he was looking for the gene for chutzpah, which he didn't succeed in finding.
Now that you've introduced humor with chutzpah, it's humorous that the identities are cloaked by.
Yeah.
Is that why you did it, just to be funny?
Norma made this slide up, who sequenced my genome. She has a very sly sense of humor, and yes, all of us, are sort of genome narcissists, and we published our genome and made it available for other people to analyze. And so she did that as a bit of a joke, because we all relinquished our privacy here.
You're angry.
So one point I would like to make that sort of gets lost in in the press around genomes is that nobody has really perfectly sequenced a human genome. Every genome that's been published to date of a human is a rough draft in one form or another. And there's a variety of reasons. They're all missing parts. The reference is not complete, and people tend to map back to the reference. There's regions that are hard to do by any technology. There's aspects of the genome that are just difficult to measure. And nobody says they've got it all perfectly right. And so, this is sort of a work in progress. The measurement tools are improving all the time. This is what we do. A lot of the research in my lab is concerned with improving ways to sequence genomes. And so it's something that's ongoing. And so one of the questions is, given that these genomes aren't perfect, is there any utility in them, or can you say anything about what it might mean for an individual's health? I want to argue that you can. And so what do you get out of it? Well, nothing good. It's all bad news. And so if you're a worrier, if a weak stomach, don't do this. That's because there's a research bias. The NIH doesn't fund research into the genetic basis for happiness and good health and things like that. They fund research into what are the causes for disease and how can we diagnose and treat them. And so when you start mapping your genome against everything that's known in the genetics literature, it starts telling you about your propensity for various diseases. But there are reasons why it's useful to know that, and I'll give you a bunch of examples. And so what we did after sequencing my genome was do sort of a provisional annotation using some software that was developed by George Church's group at Harvard, and then we enlisted my colleagues at the hospital, and the groups were led by Ewan Ashley, Russ Altman, and Atul Butte to try to do this real clinical annotation to see if having this genome in hand would be useful for my own health purposes. Now, let me give you sort of a flavor of the sort of information you get out of the genome with a few lighthearted examples, and then we can get into kind of the meat of the medical thing. So some parts of the genome are very straightforward to interpret. This turns out to be a mutation that I have in an olfactory receptor gene, which is related to how you smell. Olfaction is how you smell. And so the protein that sort of detects the odorant, one of them for me has a mutation, and this is one that's been characterized before. And turns out for most people, the hormones in male sweat, it smells rank to them and sour. Because of this mutation, it smells sweet to me, okay? And that explains a lot about my childhood I never understood, right? Didn't understand how my parents would tell me, use deodorant, and this, and it didn't make sense. Now I understand why. So some things like that, very simple to interpret. One single mutation, it's a functional thing, and it's been studied, and you understand what its role in a relation is, and cause and effect, and so forth. Other things are harder to interpret. One of the sort of famous examples is eye color. Eye color is not determined by a single gene. There's many genes involved in that, and nobody's managed to work out to completeness how eye color is determined. And so what I did for fun was I searched through the annotation and pulled out everything that had come out that was related to eye color, and I listed them all here. And if you do a majority vote here, it actually does a pretty good job. All but one are blue. And so it does a pretty good job of predicting my eye color by just a simple majority vote. And so that's one way, or a very simple approach to complex traits, is to try to collect together all the information you have from unrelated studies and set up some kind of voting scheme. And it worked here. It may not work in all cases, but it's one approach to these sorts of more complex traits. Here's another one. I stumbled across some that related to intelligence, and so I searched and collected all the genes related to IQ that have been annotated, and they all came back higher than average. And that made me feel pretty good. I was feeling very, very good about that. And then I went back and I looked at the original papers in the academic literature that were used to derive these results. And it turns out that the effect that they're measuring is so small that there's no reasonable interpretation for any individual. This question about, does this gene affect IQ, and is there an average increase of IQ? People have this gene. They can show that in a population level, perhaps that's true, but the statistics are never strong enough for you to say what the effect is on any given individual's IQ who's got the genes. And so these, to my disappointment, end up having no predictive value for me. And and there's many effects like that where people have found some interesting genetic thing, but it's only true at the population level, and it's not such a strong effect that it has a meaningful interpretation for individuals. So you have to be very careful about that. That makes sense? Okay, so what did my friends in the hospital find? It turns out that everybody's got a few skeletons in their genome. The estimates are that we all carry half a dozen or a dozen rare mutations that are deleterious in one form or another. And it would never be economically feasible to screen any one person for any given condition because it's so rare. But when you sequence the whole genome, whatever is there is laid out for you, and you can figure out what sort of hand you've been dealt. And in my case, one of the things that came out was a mutation in this protein that's involved in the heart muscles. And it was a new mutation, but other mutations in that gene have been linked to a disease called cardiomyopathy. And so the cardiologist in the group said, well, you better come to my office and we'll start to put you on the exercise bike and all that and figure out what kind of shape your heart is in, because you probably have a much higher than average risk of having a heart problem. And so fortunately, my heart is okay right now, and I did great on all the tests, and that is a relief. But for sure, this is something that I'm going to keep an eye on and check more frequently than I would otherwise. Okay, knowing that my risk for having a heart problem like this is substantially higher than the general population means I'll be tested more frequently, and this provides a way to sort of rationally allocate healthcare and testing. Right? People who are at risk should be tested more frequently. People who are not at risk could be tested less frequently, and we all have a couple things that we should keep an eye on. The genome points to, and for me, this is it. And so this was, for me, a very useful outcome, something that I'll be following over the years. And I think pretty much anyone who has the genome sequence will find one or two things like that they'll be highly motivated to keep an eye on going forward. So for common disease, heart attacks, high blood pressure, this kind of stuff, it's a much tougher problem. This is what Atul Butte worked on. And this is very similar to the eye color question, okay? If the last one was like the smell, this is more like the eye color. Where there's many studies on things that relate to heart attacks. Many genes are involved. You can work out the risks for any given gene. And the question of how you sort of integrate all that information is a challenging one. And Atul worked out something that was a little more sophisticated than that voting scheme I described, but kind of gets you the same answer. And he tried to estimate sort of my risk for heart disease. And this was, and he did this for all common disease. And he gives me a list of diseases and sort of the a priori risk, and then how the risk changes due to my genetic inheritance. You can see that risk increases for almost everything except that one. That's Alzheimer's. It's not that easy to interpret, and so he tried to find a way to put it all together. And one day you might go to your doctor and they'll give you a card like this to take home with you, where the size of the font indicates the relative risk for you. Bigger the font, the bigger the risk. And the stuff on the outside is the stuff you can affect. And again, the bigger the ball or the bigger the font, the bigger the effect is. And sort of the take-home lesson is something that you probably didn't need the genome to do. Smoking, stress, alcohol are bad. Statins, antipsychotics, diet, exercise are good, and everyone sort of knows that already. And this was maybe related to the source of Nicholas Wade's complaint that it's these common diseases are very complicated. The genetics are tricky, and everyone knows what to do anyways, right? We should all exercise more, take our antipsychotics, and so that for me was a bit of a wash. Wasn't clear how valuable it was. The third piece, though, was quite useful, and this is the work that Russ Altman's group did, and this is the pharmacogenomics, working out the sort of drug-genome interactions. And so he went through and his group, and they gave me a list of drugs. Here's the drugs right here, which, if a doctor were to try to prescribe these drugs to me, I could go and see if they're on the list and say, well, if you're trying to prescribe warfarin, you should start me off with a lower dose than someone else gets. Or if you want to give me fluvastatin, that's going to work for me. But if you want to give me these particular beta blockers, they're not going to work that well. Maybe you should try something else. And so it's a very sort of practical thing to have in hand. Again, to try to make sure that medicine you're taking is going to work for you. You're not going to waste a lot of time taking the wrong drugs, having side effects, or not having a response. And this is, for me, a very valuable outcome to the whole exercise, and something I keep in my back pocket. So the take-home lessons are that, even though the genomes are imperfect and, technology is still evolving and so forth, there are some very useful health-related insights to get out of it. But you got to beware of, sort of caveats along the way, having to do with the technical limitations, limits of the scientific literature, and stuff that just doesn't apply very well to individual interpretation, like the intelligence genes. And so you have to carry around a healthy skepticism. But it is something that I think is medically very practical that's come out of the whole exercise, and it wouldn't have been possible to do without the reference genome in the Human Genome Project. And now numerous companies are springing up both to do the sequencing and give this interpretation to people. The genomic data is growing exponentially, and it's really quite amazing how fast it's taken off. So that's sort of the most obvious thing to do with the genome is, everyone gets their own genome, tries to figure out what it means to them. There are numerous other ways that the genome is being used now for clinical purposes, and I want to describe one or two in the remaining time. So one of them has to do with making babies. And humans are supposed to have 2 copies of each chromosome, okay? And this is called a karyotype. This is some individual's sort of genome laid out. 2 copies of chromosome 1, 2, 3, so forth. You can see that this individual has 3 copies of chromosome 21, which is not a good thing. And it's more commonly known as Down syndrome. And this is the most common genetic disorder for babies. This kind of Down syndrome, number 1 for sure, having an extra copy of chromosome 21, but not having the right number of chromosomes more generally is called aneuploidy, and they affect about 1% of live births. And there are ways to measure that before the baby's born. The problem is that they involve risk. So you take a big needle, stick it in the mom's belly, right up next to the baby, and try and get a few of the baby's cells. The challenge is trying to get the genetic material. And so that causes a risk both for the mom and the baby. And when I became a parent and saw this done to my wife and our unborn daughter, and then a few years later this done to our future son, it was really disturbing, and it's just seemed awful that you had to risk the pregnancy to ask a question. And so I got interested in trying to develop a non-invasive method to make genetic measurements, and sort of get risk out of the equation. And I was sitting in my office one day flipping through a journal and discovered a reference to this very old literature dating back to the late 40s, where these 2 Frenchmen discovered that there is DNA in your blood that's not part of any cells anymore. So the DNA, I said at the beginning, is the blueprint for making all the constituents of the cell, and it sits there in the nucleus of the cell. It's wound up on spools and all protected, and occupies this sort of very privileged place. And that's true, but it turns out that when a cell dies, the genome doesn't go to heaven. It gets dumped into the blood and chewed up into little pieces. And so it's naked bits of DNA that'll circulate in the blood that are sort of the detritus of dead cells. And that's what these guys discovered in 1948, that there is DNA outside the cell. It just floats around in the blood. And it was kind of very obscure and forgotten about for a long time. And then 50 years later, these guys in Oxford discovered that for pregnant women, some of that DNA in the blood comes from the baby. So, and they did that by looking at the sex chromosomes. And so, if you're a woman, you're XX. If you're a man, you're XY. So women should not have any Y chromosome DNA anywhere. Turns out if you're pregnant and carrying a male fetus, you have some Y chromosome DNA floating around in your blood. That's what they showed in the study. Therefore, it must come from the baby. And that set off a sort of decade-long race to try to use this as a window in the genetics of the fetus. And what we did is work out a way of using sequencing and the reference genome to work out if the baby had an extra copy of a chromosome or not. And this was work done by Christina Fan, who was a student at the time in my lab, and our clinical collaborator, Yair Blumenfeld, at the hospital. And the basic idea is that if you look in the mom's blood, the mom is putting in 2 copies of chromosome 21. She's healthy, normal, diploid person, and 2 copies of every other chromosome. Let's say chromosome 1 is the control. The baby's putting in 3 copies. If the baby's got Down syndrome, the baby's putting in 3 copies of chromosome 21 for every 2 copies of chromosome 1. So there should be a very slight excess of chromosome 21 DNA in the blood relative to chromosome 1. And if you could count these molecules up and figure out what chromosomes they came from, you could in principle measure that very small difference. And so that's what we did. We used a sequencer to count molecules and to work that out. The experiment is pretty simple. We did a study in the Stanford hospital in which we drew blood from pregnant women, some of whom were carrying babies with Down syndrome. Christina purified the DNA from the blood, and she sequenced it on one of these next generation sequencers. And she sequenced a lot of molecules, about 5 million molecules per patient sample. Every single one of those little molecules we mapped back to the reference human genome to try to figure out which chromosome it came from. And we allowed it to vote for a chromosome based on that. And then we looked for overrepresentation of any given chromosome. Sort of like looking for voter fraud. Every molecule votes for a chromosome. We're looking for someone that's overrepresented. And this shows the result for chromosome 21. On this graph, it shows sort of the relative representation relative to the other chromosomes. And the red data points are the women whose babies had Down syndrome. The blue data points are the women whose babies didn't. And you can see that the Down syndrome runs are all overrepresented in chromosome 21. You can draw a line that separates the data. It's clear as day. That makes sense? Okay. And it turns out that there's nothing special about chromosome 21 in this measurement because we're just randomly sequencing. We get the voting for all the chromosomes, and we can detect other genetic diseases like trisomy 18. These 2 babies had that. Trisomy 13, that one had that. And just read it all off. And so these are the 3 survivable trisomies. So these are the 3 that matter for live births. So we published this in 2008. And it set off sort of a real burst of activity and over the next several years. And this year and the year before, several large-scale blinded clinical trials published. Thousands of women have been tested with this approach, and it worked exactly the way we said it would in our first paper. And so this is now something that's available in the clinic for people to use. And it's been sort of an amazingly fast sort of transition from basic university research to something that's practically available and a very sort of clear product of the Human Genome Project. I think I'll maybe say one more example and then we can stop and talk. And this one uses the same idea of looking at cell-free DNA, the DNA that circulates in the blood, but for a completely different medical condition, which is organ transplantation, in this case heart transplantation. So it turns out, well, after we published our work on the prenatal diagnostic, word got around Stanford that I was interested in non-invasive diagnostics, and I got a call one day from Hannah Valentine, who's a cardiologist, and she says, we have this horrible problem in heart transplants, and can you help us solve it? It turns out that when you get a new heart put in, it's a major operation, sometimes your immune system will attack the new heart and you reject the transplant. And so the doctors try to treat that with immunosuppressants, but you take drugs that suppress your immune system, you'll get sick very easily from bacteria and viruses and so forth. That's not a good thing either. And so they give you the minimal amount of drugs, but then they try and check you for rejection. And they do that by going in and physically ripping out a piece of your new heart every couple of months. They go in and physically biopsy it. Yeah, it's dangerous, it's painful, it's expensive, and it's actually not a very good diagnostic. And so we decided to try to fix this problem, and this is the work that was done by my then postdoc, Tom Snyder. And the idea here is to think about the heart transplant as a genome transplant. Okay, you're getting someone else's genome put in your body. All the cells in that heart have someone else's genome. It's different than yours, and we can measure those differences. So before the transplant, imagine your genome and all the mutations. Remember, there's about 2-3 million mutations that are special to you. Let's call them red mutations, and all your DNA in your blood is red. It's coming from all your organs. Now you get a heart transplant from somebody who's got a green genome. And so any cells that die from the heart, they're going to contribute green DNA molecules into the blood. And if your immune system starts to attack the heart, more cells die, more DNA from the heart gets thrown in the blood. That should increase the amount of green DNA in the blood. Okay? And so this is kind of a way to measure damage to the heart by looking at the relative amount of foreign DNA in your blood or foreign mutations. And so again, we do this by sequencing. We sequence a lot of cell-free DNA. We measure ahead of time, not the whole genome, but a large number of mutations in both the donor and the recipient. And we can then monitor the percentage of donor DNA in the blood. And sure enough, these graphs are all different patients. Don't worry about the green lines. The blue lines are the percentage of donor DNA as a function of time. And at every data point, these patients were also physically biopsied, and a pathologist gave a score for healthy or rejecting. And all the scores were healthy, except for the ones where there are red arrows pointing to it. Okay, and so you can see that the cell-free DNA does a great job of predicting rejection. You know, it shoots up not just at rejection, but in many cases before rejection. So it predicts rejection much earlier than the biopsies do. We think this is going to be sort of a very powerful diagnostic that's going to be safer, less costly, and lead to better outcomes for patients than conventional practice. Again, sort of a direct consequence of the Human Genome Project. And in this story, there's nothing I've told you that's specific to the heart. Should work for any organ. Heart is actually very small by mass compared to lungs and liver, and so we're now following on studies to try and prove its generality in all organs. So that's been sort of the work we've done in developing sequencing technologies and trying to understand the human genome. I'm going to take a detour right now into sort of the other part of my life that Lynn mentioned, which has been to try to create biological automation and small plumbing. And so we've worked very hard to try to create chips that are sort of the biological equivalent of the integrated circuit. And so, you know, your computer chips have transistors and wires and move electrons around. We have chips that have pipes and valves and pumps, and we can push fluids and molecules and cells around. And we can do this with now a pretty high degree of complexity, up to really tens of thousands of valves on a chip. And with that in hand, we began looking at ways to attack new problems in biology. And you know, one of the first ones we looked at was structural biology, which is how you make pictures of proteins and work out their atomic scale structure. This ends up being very important in the drug industry, for example, as well as for sort of basic biological studies. And I'll sort of make a long story short by saying we worked out a very powerful way to crystallize proteins and get their structures. This was the work of a student named Carl Hansen. It was commercialized through a company I founded called Fluidigm, and X-ray crystallographers around the world started to use it and work on problems that really matter for human health. Here was one that solved the structure of a protein Ebola virus. There's one for H5N1 flu. Let's see, where'd that go? Right here, by Ian Wilson's group, and another one by Tim Springer looking at integrins and fibrinogen mimics, so drugs related to cardiovascular disease. And so it's been a way to see our research get in the hands of people around the world. The company performs a really sort of useful function there. We've also developed chips that allow us to measure molecular interactions. We've used this for basic science. We've used it for drug discovery. Again, it's the sort of thing that's finding its way into labs all over the place and has become a very powerful part of biological measurement. And I'll just kind of try and wrap up by saying we've now begun to fuse these two parts of our research and combine high throughput genomic sequencing with the small plumbing and use this to study genomes from single cells. And there are a lot of good reasons to do this. So one is to look at microbes, little bacteria and such. Turns out that only about 1% of bacteria can be grown up in a laboratory. 99% of what's out there you can't grow. And in conventional microbiology, you've got to grow it to study it. And so we got interested in trying to explore all this microbial diversity, which matters for environmental issues. This particular one is involved in red tides and things like that, keeping the ammonia balanced in the oceans. We've been interested in looking at bugs that matter for human health. This TM7 is one that lives in everybody's mouth and is related to oral disease. And so there's a whole bunch of the vast majority of the microbial universe which is important in the environment and in health, but which can't be accessed by conventional microbiology. And these tools are providing a way to get at that. And so that's been kind of a nice fusion of the two flavors of research we've done. And we've most recently taken it to now doing single molecule, single cell human sequencing, in part to try to address some of those technical challenges I mentioned about getting genomes to be a more faithful representation of reality. And I won't dwell on the technical details of that, but this has been an important step. I mentioned that we had started a company to make this plumbing called Fluidigm, and they have created about 100 jobs in the US. Half the companies in the US, half's in Singapore. And there's very interesting stories behind that if you're interested in the policy side of that. They announced that last year that they had built and shipped 1 billion valves, which is equivalent to all the conventional plumbing in every home in California, Texas, and New York. And you know, I know there's a lot of concern about job outsourcing. I'll tell one little vignette about this. That at a board dinner, I was talking to the CFO. They've been thinking about opening a second factory, and he said, you know, it's not. They've been thinking about whether to do it in the U.S. or abroad, and he said, you know, in the U.S. We could do it, and the salaries would be about the same as in Singapore. There's not really a salary difference. Where the big difference in cost comes in is in the healthcare costs for the employees, and so I found that to be quite interesting. Skip that, and I'll just go to the conclusions, and we can have a discussion if you like. I hope I've been able to convince you that there are some really practical applications of the human genome, practical for medical purposes in ways that are going to improve people's health and lower costs of healthcare. And you know, it's been sort of a really powerful thing to have in our scientific arsenal, to have the human genome and to be a part of developing these sequencing technologies. And of course, to have the support of the government in doing this. All the research I've described here that's been done in my lab has been, virtually all of it has been funded by the government, primarily by the NIH and by DARPA. Things like the NIH Director's Pioneer Award, the NHGRI, and the ARA Act have all played an important role in this research, and we couldn't have done it without that support. So with that, maybe I'll stop here and we can have a discussion, questions, whatever you like. Yeah.
What are the barriers to especially with the heart transplant? What's the barrier to that becoming common practice?
The diagnostic becoming practical. Yeah, so you need to do some clinical studies. You need to do the blinded prospective studies, and that's something that we're trying to get funded now.
Is like that method that you were talking about, are you familiar with AlloMap? So is that the same thing? Or?
No. Yeah, so at AlloMap is sort of a failed diagnostic. What they were trying to do was look at gene expression in the immune system. So they developed an assay that sort of tells them that the immune system is activated, but it doesn't distinguish between being activated because you're attacking this foreign heart or being activated because you're attacking a bacterial infection. And so it's a different sort of measurement, one that's not a direct measurement of the health of the heart.
So that method is more complete analysis.
Yes. The thing we've done is a much better predictor of rejection. Yep.
I think that whole process of sequencing a person's whole genome and using it as kind of a preventative healthcare system, do you think that can become widespread, that babies born, we're going to have the sequencing of the genome? Do you think that can eventually be done?
Yeah, it's going to happen. We're not that far away from the $1,000 genome, and so let's say it does get to $1,000. That sounds like a lot of money, but you average it out over the cost of your life. It's what is it? It's $10 a month for 10 years, and so it's pennies a day for the first 10 years of your life, and then you're done, and it's by the time you're 10 years old, it's complete. And so and because of this nature of being a one-time measurement, I think it's not that expensive, and I think you'll be able to make the health economics cases that the benefits you get out about what to screen for preventatively, and how to properly take drugs that without causing side effects will be strong enough that there'll be a pretty compelling economic case for doing that. Yeah.
What do you think needs to happen in order for the information that you had shown us about the different drug reactions and different risk factors for diseases? What do you think needs to happen for that to sort of be incorporated into patient EHRs?
To be incorporated into
patient EHR.
Yeah.
So that when they do go see their doctor, everything
Yep.
is in their file.
Yep. Well, we got to have patient EHRs first, I think. Probably I don't think there's a huge technical gap right now. The sequencing part, solved problem. It costs a certain amount now. The cost is going to go down over time. The annotation of it right now is fairly labor intensive. There were only 4 authors on my genome paper, but there are 30 on the annotation paper. So but that's something that many people are working to solve. In another couple of years, that's going to be pretty straightforward. And then, the missing gap is going to be how to get it in the hands of the doctors. And the doctors, they aren't very well educated about genomics in general, and so there's a curve there. And I think you'll see an increasing role for genetic counselors, and huge increase in the number of genetic counselors needed for sure.
That leads to my second question about maybe some policy implications in terms of how you then give that data back to a patient. You are a scientist, you know what you're looking at. You have a lot of people that might be able to spend $1,000 on their genome, but then don't really know what to do with that information. I feel like there needs to be some sort of policy guidelines in place as to how that information is delivered, and some of that will be educating physicians on.
Yep. And so genetic counseling, there's a huge infrastructure that needs to be put in place. That's probably the rate limiting step, to be honest. Yep.
I have 2 questions. One is, a pregnant woman can now request this.
Yep.
And my other question is, if all of a sudden you had to go out into the private insurance market and buy health insurance for yourself, would the fact that your genome has been sequenced have any impact? I mean, could an insurance company ask you if your genome has been sequenced, and if so, could they ask to see it? Could they uprate you because of your, tendency to cardiomyopathy, that whole thing?
Well, so the GINA Act makes it illegal to discriminate based on my genetic information, but it's still totally legal to discriminate based on my phenotype. And so, as a point in fact, a couple years ago, my dad was bugging me to go get more life insurance, take care of my, started a family. So you need more life insurance in case anything happens to you. And I, called up the life insurance company. The guy came to see me, and he wanted me to take a bunch of tests, blood test, echocardiogram, this and that. And if any of those didn't come out the way they wanted, they could have denied me coverage. And so, discrimination is rampant in the insurance business based on things other than the genome, which they view as proxies for it. And that's sort of a major issue, I think.
So GINA covers health insurance?
I'm sorry?
GINA covers health insurance?
Yeah. Yep. That was the point. Yep.
So with the personal screening, it seems like that raises, has there been any research on the psychological, aspects of knowing, because obviously you're very familiar with the work, but most of the people when it becomes commercialized aren't going to be as comfortable with what this data is actually telling them. Has there been any research?
Yeah. So two answers to that, I think. On the level, we'll talk about both for that and for the prenatal thing. Okay. So for the genome analysis, I think there's a certain fraction of the population out there that wants to know. I was in that group. I want to know. I feel like I'm making a lot of decisions that, if I knew I was going to die next year, I would change what I'm doing today. And so I was very curious to know. But there's a lot of people who just don't want to know. I mean, for whatever reason, it's not interesting to them, the decision they make. They're feeling divorced from that. And you can ask, what's the relative proportion of those two kinds of people? And it's about 50-50. And the way you know that is through well-studied diseases like Huntington, okay, which is sort of as close to a genetic death sentence as you can get. You can go in and make a measurement of one gene, look at the repeat length there, and it's a very good predictor of when you're going to die. And people who are at risk end up getting offered this test, and about half of them decline. They just say, I don't want to know, even though it's an incredibly specific predictor of their end of their life. And you have to respect that, I think. That's human nature. Half and half. The other one is on the prenatal one, which is one that is sort of a challenging one because it's often linked to termination decisions. And but there have been studies that show for people who decide they want to carry their baby to term with Down syndrome and not terminate, the earlier they know they've got a baby with Down syndrome, the better prepared they are to deal with the challenges of raising a kid with special needs. And so the outcome, having a safe early test leads to better outcomes regardless of the person's decision about termination or not. And so that's known as well. Yeah.
Either you or your colleagues have looked at how an environmental exposure and various, how a particular gene might react differently or cause a different reaction to an environmental exposure?
Well, I mean, there's a whole field of epidemiology is concerned with that, and it's not something that is sort of my personal area of expertise. But again, there's that all filters into these databases that the genome gets mapped onto, and genes that are linked to higher sensitivity environmental exposure might pop up for some people. It didn't for me.
Because a drug is just a chemical like it carries pollutants and nothing more than a chemical. So science would be the same.
Yeah.
So you alluded to the idea that earlier, that maybe your prenatal test could be done earlier than the more invasive. Mm-hmm. Is that in fact the case? Mm-hmm. Is it so it's equally accurate earlier in pregnancy?
Equally accurate as an amniocentesis? Yeah. Mm-hmm. Right. Yep, it is. Because amniocentesis is not a very accurate test. Yeah, it's doctors, they try to hide that, but when you really dig down, it turns out it's really, yeah. Frightening. Yeah, exactly. Yeah.
Going to eye color, if there's a baby that's just born, will you be able to test maybe see what color their eyes will change to if the baby's X gene?
Yeah, so that's really an interesting question. And one thing I didn't talk about that we've been doing right now has been, we can do the whole baby's genome prenatally before they're born. And that opens up all kinds of interesting questions. First of all, can you interpret it? You could try the voting thing for eye color, maybe you could. I think, my own sense is that it's going to be less useful than the Down syndrome test, just because the Down syndrome thing is the most common thing you would think about. But there are things that the genome is useful for, and I'm thinking it's going to play a role in the third trimester, late in pregnancy, to discover genetic conditions that you want to treat immediately after the baby's born. If you've been a parent, it's total chaos the first couple of weeks there in the hospital. And so there's all kinds of metabolic and immune disorders that you'd want to know right away. So you would feed the baby the right food, PKU, maple syrups, and this kind of stuff. You don't want to find that out the hard way. Or if your baby has got immune disorder, and it'd have to be in a sterile environment, you want to know that ahead of time so you're ready. And so I think that'll be the value of the prenatal genome sequencing. It's figuring out conditions for which you'd want to treat the baby immediately when they're born. And in the more science fiction world, maybe before they're born. That's more science fiction right now. Yeah?
Would this be applicable in the IVF situation, the implantation genetic diagnosis?
Yes. Yep, for sure. Although very small fraction of babies are made that way. Most of them are made the old-fashioned way.
More fun.
Yeah.
So if you get the baby tested right when they're born, I don't know anything about DNA, but does it change when they get older? Would you have to get them tested at all?
Yeah.
Or is that one test enough?
It's actually a very subtle issue, but speaking very generally, one test is enough. Yeah, you've got one genome and you've got it all your life. till it mutates and gives you cancer. But that's a whole nother story. Yeah, thank you.